Carbohydrate is one of the major elements of food that we consume. As a medical student, you need to have a crystal clear conception on carbohydrate metabolism. In this video, we are gonna have a detailed discussion on this topic. Well, we all know metabolism have two parts. One is anabolism and another is catabolism. In carbohydrate metabolism, glycogenesis and gluconeogenesis is anabolic pathway, and glycolysis, glucogenolysis, HMP shunt, oxidation of pyruvate to acetyl-CoA and TCA cycle is catabolic pathway. After absorption of glucose, it maintains glucose pool, which is total amount of glucose present in ECF. This glucose is oxidized through glycolysis and a little portion is oxidized through HMP shunt. Some portion of glucose is turned into glycogen and some portion in non-essential amino acid and some portion is in fatty acid and fat. Now we're gonna have a look on glycolysis. In aerobic glycolysis, the glucose is converted to pyruvate, and in anaerobic glycolysis, the glucose is converted into lactate. Here, the rate limiting enzyme is phosphofructokinase. The ATP production. In aerobic state, 8 ATP is produced per glucose, and in anaerobic state 2 ATP is produced. We will calculate this ATP production later. Now we will see the steps of glycolysis. At first, glucose produces glucose 6-phosphate with presence of hexokinase or glucokinase and magnesium ion, and here ATP is converted to ADP. In next step, glucose 6-phosphate produces fructose 6-phosphate with presence of phosphoglucoisomerase. Next, breaking this ATP into ADP and inorganic phosphate, fructose 6-phosphate produces fructose 1,6-bisphosphate with presence of phosphofructokinase. Now, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate is produced from fructose 1,6-bisphosphate with the presence of aldose enzyme. With the presence of dehydrogenase enzyme, glyceraldehyde 3-phosphate produces 1,3-bisphosphoglycerate. Next, 1,3-bisphosphoglycerate produces 3-phosphoglycerate with presence of phosphoglycerokinase enzyme and magnesium ion. Here 1 ATP is produced. In next step, 3-phosphoglycerate is converted into 2-phosphoglycerate with presence of mutase enzyme. Then this 2-phosphoglycerate is converted into phosphoenol pyruvate with presence of enolase enzyme. This phosphoenol pyruvate produces pyruvate with presence of pyruvate kinase and magnesium ion and producing 1 ATP. Well, we found 1 mole, 1 atom of dihydroxyacetone phosphate and 1 glyceraldehyde 3-phosphate. Here we can see this dihydroxyacetone phosphate can be converted into glyceraldehyde 3-phosphate. So, following this pathway, this dihydroxyacetone phosphate can produce 1 pyruvate. In this way, glyceraldehyde 3-phosphate produces 1 pyruvate and dihydroxyacetone phosphate produces 1 pyruvate. Up to this step, the pathway is for aerobic glycolysis. So, we can see 1 molecule of glucose produces 2 pyruvate in aerobic glycolysis, which we have told before. In anaerobic glycolysis, pyruvate produces lactate with the presence of lactate dehydrogenase. Here, one molecule of NAD2H is used, which was produced in this step, where glyceraldehyde 3-phosphate is converted into 1,3-bisphosphoglyceraldehyde. Now we will count the produced ATP both in aerobic glycolysis and anaerobic glycolysis. Here in this two-step, 2 ATP is produced, and here 1 NAD2H is produced. This NAD2H can produce 3 ATP in electron transport chain. So we can see one molecule of glyceraldehyde 3-phosphate can produce 5 ATP. Here, as this dihydroxyacetone phosphate follows this pathway and it also makes 5 ATP. So totally 10 ATP is produced in aerobic glycolysis. But in first and third step we can see 2 ATP is used. So net ATP production is 8 ATP in aerobic glycolysis. On the other hand, in anaerobic glycolysis, this NAD2H is used. So only 4 ATP is produced, where in first and third step 2 ATP is used. So net production in anaerobic glycolysis is 2 ATP. Now we will learn the importance of glycolysis. It is the principal route for glucose oxidation to generate ATP. It is also the pathway for fructose and galactose oxidation. Moreover, it is the only metabolic pathway that can continue to provide ATP even in anaerobic state. Here in RBC, anaerobic glycolysis takes place as RBC have no mitochondria. So, through anaerobic glycolysis, RBC produces ATP, which maintains biconcave shape of RBC. Mainly, this produced ATP in RBC is used to maintain the activity of sodium-potassium pump, which pump maintains the shape of the RBC. In RBC, 2,3-BPG is produced by BPG shunt. This 2,3-bisphosphoglycerate is required for hemoglobin to transport oxygen. Here comes the BPG shunt, which takes place in RBC. Normally, glucose produces 1,3-bisphosphoglycerate, and this 1,3-bisphosphoglycerate produces 3-phosphoglycerate. But in RBC, through BPG shunt, 1,3-bisphosphoglycerate produces 2,3-bisphosphoglycerate, then 2,3-bisphosphoglycerate produces 3-phosphoglycerate, and this 2,3-bisphosphoglycerate helps hemoglobin to carry or transport oxygen. Now we will discuss on oxidation of pyruvate to acetyl-CoA. It takes place in the mitochondria of all cells. In this process, some coenzyme is needed, like thiamine pyrophosphate, NAD, FAD, coenzyme A, lipoic acid. Here 3 ATP is produced for each pyruvate. Now we will see the chemical reaction by which pyruvate is converted into acetyl-CoA. Here 2 enzymes is needed. One is pyruvate dehydrogenase and another is thiamine pyrophosphate. Here NAD is reduced and one molecule of carbon dioxide is removed. Now we will talk about TCA cycle. Here the substrate is acetyl-CoA. The rate limiting enzymes are citrate synthase, isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase. And 12 ATP is produced for each mole of acetyl-CoA. Here is the steps of TCA cycle. From the oxidation of pyruvate, acetyl-CoA is produced. This acetyl-CoA joins with oxaloacetate and produces citrate with the presence of citrate synthase enzyme. This citrate produces isocitrate with aconitase. This isocitrate produces alpha ketoglutarate by the presence of isocitrate dehydrogenase. Here, one molecule of carbon dioxide is removed and one NAD molecule is reduced. In the next step, alpha ketoglutarate produces succinyl-CoA, and also in this step, one molecule of carbon dioxide is removed and one molecule of NAD is reduced, and in this step, alpha ketoglutarate dehydrogenase enzyme acts. In the next step, this succinyl-CoA produces succinate with the presence of succinate thiokinase, and here one ATP or GTP is produced. Next, this succinate produces fumarate with the presence of succinate dehydrogenase, and here one FAD molecule is reduced. Then this fumarate produces malate with the presence of fumarase enzyme. This malate produces oxaloacetate. Here malate dehydrogenase enzyme acts and one molecule of NAD is reduced. Teacher may ask you to justify that oxaloacetate is the catalyst of TCA cycle. We'll learn to justify it. Literally, acetyl-CoA consists of two carbon molecules. Actually, this acetyl-CoA is lost through the cycle as two molecules of carbon dioxide. After the removal of two carbon dioxide molecule, we can find 4 carbon molecule containing succinyl-CoA. Here, the oxaloacetate is also 4 carbon molecule containing particle. So, through the successive steps, succinyl-CoA produces oxaloacetate, which is 4 carbon molecule containing group. So we can say oxaloacetate is reproduced through the successive steps, and this oxaloacetate again and again continues this cycle. So we can see, reutilizing oxaloacetate, a small amount of oxaloacetate oxidizes a large number of acetyl-CoA. For this reason, oxaloacetate is called catalyst of this TCA cycle. Now we will count the ATP produced in this TCA cycle. Previously in our discussion, we told that 12 ATP is produced for one molecule of acetyl-CoA. Here we can see the production of 3 reduced NAD. This 3 NAD in electron transport system can produce 9 ATP. On the other hand, we can see one molecule of reduced FAD is produced. In this electron transport system, this reduced FAD can produce 2 molecules of ATP. And there is 1 ATP which is produced in this TCA cycle directly. So we can learn that totally 12 ATP is produced through this TCA cycle for one molecule of acetyl-CoA. another important topic related to this TCA cycle which you may face in your exam hall. Examiner may ask you to justify that TCA cycle is an amphibolic pathway. Amphibolic pathway means it has both catabolic and anabolic parts. In TCA cycle, acetyl-CoA is oxidized to produce ATP and carbon dioxide, which is catabolic part of TCA cycle. On the other hand, we can see that in anabolic part, some important intermediates of TCA cycle like citrate, alpha-ketoglutarate, succinyl-CoA, oxaloacetate, this can produce some important elements. Citrate in cytoplasm gives acetyl-CoA for synthesis of fatty acid, cholesterol and steroid. Alpha-ketoglutarate can be used for synthesis of glutamate and glutamine. Succinyl-CoA can be used for protein and heme synthesis. From oxaloacetate, aspartate and aspargine can be synthesized. Even many of these intermediates can take part in gluconeogenesis. You may have also asked to prove that TCA cycle is a common metabolic pathway. Now we will learn to justify that TCA cycle is a common metabolic pathway. It is said to be a common metabolic pathway because elements from carbohydrate, fat and protein enters into TCA cycle. Glucose from carbohydrates is converted into pyruvate and this pyruvate produces acetyl-CoA, which acetyl-CoA enters TCA cycle. On the other hand, fatty acid and glycerol comes from fat. This glycerol produces pyruvate, which pyruvate produces acetyl-CoA and this acetyl-CoA enters TCA cycle. On the other hand, fatty acid produces acetyl-CoA, which acetyl-CoA enters TCA cycle. In the case of protein, glucogenic amino acid and ketogenic amino acid can be produced. This glucogenic amino acid produces pyruvate and this pyruvate produces acetyl-CoA, which acetyl-CoA enters TCA cycle. And on the other hand, ketogenic amino acid directly produces acetyl-CoA and this acetyl-CoA enters TCA cycle. Now we will count the total ATP produced in the complete oxidation process of one molecule of glucose. In glycolysis, we saw one molecule of glucose produces 8 molecules of ATP. And from conversion of pyruvate to acetyl-CoA, 6 ATP is produced. And in TCA cycle, each acetyl-CoA produces 12 ATP. So, from glucose, we found 2 acetyl-CoA. So, we can say glucose can produce 24 ATP in TCA cycle. Totally, we can find 38 ATP from complete oxidation of one molecule of glucose. Now we will learn the importance of TCA cycle. Firstly, it oxidizes metabolic fuels to provide reduced coenzymes. Then we learn that it is the final common metabolic pathway. We also learned that it is an amphibolic pathway. TCA cycle provides some precursors for synthesis of protein and nucleic acid. Lastly, it provides carbon dioxide and ATP, which is needed to continue urea cycle. Now we will talk about HMP shunt, which is a very important topic for both written and viva examination. Here we can see that HMP shunt occurs in the cytoplasm of liver, adipose tissue, RBC, gonads, adrenal cortex, and macrophage. Here, the rate limiting enzyme is glucose 6-phosphate dehydrogenase. You can see a very big cycle of HMP shunt, but you don't need to memorize that cycle. You need to have a overall conception of the reactions of HMP shunt. Here, at first, 6 molecule of glucose 6-phosphate produces 6 molecule of ribulose 5-phosphate and 6 molecule of carbon dioxide, and 12 NADP is reduced. Then this 6 molecule of ribulose 5-phosphate produces 4 molecule of xylulose 5-phosphate and 2 molecule of ribose 5-phosphate. Here, xylulose have 5 carbon and ribose have 5 carbon also. 4 xylulose molecule have 20 carbon and 2 ribose molecule have 10 carbon. Totally, this 6 molecule have 30 carbon. In the successive reactions, 5 molecule of fructose 6-phosphate is produced, where fructose have 6 carbon and 5 fructose contains 30 carbon. Then this 5 molecule of fructose 6-phosphate produces 5 molecule of glucose 6-phosphate. Literally, this reaction was started with 6 molecule of glucose 6-phosphate and ends with 5 molecule of glucose 6-phosphate. So we can see that 1 molecule of glucose 6-phosphate is lost through 6 molecule of carbon dioxide. You may be asked to answer the importance of HMP shunt. We see that HMP shunt provides ribose, which can synthesize nucleotide and nucleic acid. This HMP shunt provides reduced NADP, which helps in production of fatty acid, steroid, and cholesterol, helps in detoxifying function of liver to convert toxic substance into non-toxic forms, prevent hemolysis of antioxidant activity in RBC, facilitates superoxide and free radical production in phagocytes to kill bacteria and other pathogens. Now we will see how NADP2H helps phagocytes to kill the bacteria and other pathogens. Phagocytes consumes oxygen, which produces superoxide with the help of NADP2H. This superoxide produces hydrogen peroxide. This hydrogen peroxide produces hydroxyl radicals and hypochlorous acid. Both the hypochlorous acid and hydroxyl radical kills the bacteria. Now we will learn that how hemolysis is prevented by antioxidant activity in RBC. We know that RBC contains hemoglobin which carries oxygen. This oxygen can produce superoxide and hydrogen peroxide. This hydrogen peroxide can produce hydroxyl radical which can damage or lysis the RBC membrane. But by the help of NADP2H, this hydrogen peroxide can be converted into water. On the other hand, hemoglobin can be converted into methemoglobin while carrying oxygen. This methemoglobin also converted into hemoglobin by NADP2H, which was produced through HMP shunt. Now we will talk about gluconeogenesis. Through gluconeogenesis, non-carbohydrate substrate produces glucose. Here the substrates may be glucogenic amino acid, glycerol, pyruvate, lactate, and intermediates of TCA cycle. Mainly gluconeogenesis occurs in liver, and the other site where gluconeogenesis takes place is kidney. Here, the rate-limiting enzymes are pyruvate carboxylase, phosphoenol pyruvate carboxykinase, fructose 1,6-bisphosphatase, and glucose 6-phosphatase. In gluconeogenesis, NADH is needed as coenzyme, and 6-ATP is needed to produce one molecule of glucose from 2-pyruvate or 2 molecule of lactate. These steps of gluconeogenesis may seem to be very difficult to you, but basically it's very easy. Actually, it is the combination of glycolysis, TCA cycle, and some relevant accessory reactions. In glycolysis, you may have noticed that there is 3 reactions which are one way in direction in optimal body condition. They are this reaction, this, and this. So you can see in glycolysis, glucose can easily be converted into glucose 6-phosphate in optimal body temperature and optimal body condition. Then fructose 6-phosphate also can convert it into fructose 1,6-bisphosphate, and phosphoenol pyruvate can also be converted into pyruvate. But glucose 6-phosphate cannot be converted into glucose. Fructose 1,6-bisphosphate cannot be converted into fructose, and pyruvate cannot be converted into phosphoenol pyruvate. So, to produce glucose from glycerol, lactate, pyruvate, glucogenic amino acid, and many other intermediates of TCA cycle, we need to produce glucose from glucose 6-phosphate, produce fructose 6-phosphate from fructose 1,6-bisphosphate, and phosphoenol pyruvate from pyruvate. Here, 4 enzymes can help us to alter the direction of this reaction or alter the process. Pyruvate cannot be directly converted into phosphoenol pyruvate. To produce phosphoenol pyruvate from pyruvate, we need to produce oxaloacetate from pyruvate first. Here, pyruvate carboxylase enzyme converts this pyruvate to oxaloacetate. This oxaloacetate can be converted into phosphoenol pyruvate by phosphoenol pyruvate carboxykinase. So, our one problematic step is solved. Now, to produce fructose 6-phosphate from fructose 1,6-bisphosphate, fructose 1,6-bisphosphatase enzyme can help us. And at last, glucose 6-phosphatase enzyme helps us to convert glucose 6-phosphate into glucose. So, we have overcome all the 3 steps. Rest of the reaction is very easy. Glycerol is first converted into glycerol 6-phosphate, then it is converted dihydroxyacetone phosphate, which dihydroxyacetone phosphate along with glyceraldehyde 3-phosphate makes one molecule of glucose. On the other hand, lactate. This lactate is converted into pyruvate, and this pyruvate enters into inverse glycolysis reaction. Here, glycogenic amino acid can enter this pathway by producing pyruvate or oxaloacetate or fumarate or alpha-ketoglutarate or succinyl-CoA. Firstly, this intermediates of TCA cycle is converted into acetyl-CoA and then enters the inverse glycolysis reaction. Now, we will learn about Cori cycle. It have a gluconeogenic process. We know in anaerobic glycolysis, glucose produces lactate. This lactate through blood circulation enters liver, and in liver this lactate produces pyruvate, and through gluconeogenesis it produces glucose, and which glucose releases into blood circulation. So, Cori cycle is integration of anaerobic glycolysis and gluconeogenesis. It prevents lactic acid accumulation, which prevents lactic acidosis. And it also reconverts glucose, which again goes to peripheral tissues for oxidation. Now we will have a discussion on glucose-alanine cycle. Here alanine is an amino acid. This alanine can be produced in muscle. In muscle, glucose produces pyruvate. In transamination process, pyruvate produces alanine binding with amino group. This alanine then exported to liver. In liver, alanine again reconverted into pyruvate, and then this pyruvate produces glucose. In liver, this process by which alanine produces glucose is a gluconeogenesis. Now we will talk about glycogenesis. Glycogenesis is a process by which glycogen is formed from glucose. Here the rate limiting enzyme is glycogen synthase, and to produce glycogen 3 ATP is needed for adding each glucose. In this process, at first glucose produces glucose 6-phosphate, then glucose 6-phosphate produces glucose 1-phosphate. This glucose 1-phosphate produces UDP glucose binding with UTP and releasing pyrophosphate. Here we need a primer. This primer molecule accepts UDP glucose. From this UDP glucose, the glucose molecule is added to the primer. This process repeats. So, a new UDP glucose molecule comes and glucose molecule adds to the primer or the previous precursor. Here, the branching enzyme helps in branching. Another process of carbohydrate metabolism is glycogenolysis. The mechanism of glycogenolysis is not that much important, so we will skip the mechanism here. We will learn only about the difference between muscle glycogenolysis and liver glycogenolysis. At first, we need to know what is glycogenolysis. Glycogenolysis is the process by which glucose is produced from glycogen. Here we can see that in muscle glycogenolysis, major product is glucose 1-phosphate. And in liver glycogenolysis, major product is free glucose. Here the liver provides free glucose from liver glycogenolysis, but muscle cannot provide free glucose. They provide glucose 1-phosphate. Glucose 1-phosphate is not capable of coming out of the muscle cell, but the free glucose produced in the liver can enter the blood circulation. So, glucose produced in liver can maintain the blood glucose concentration, but the glucose produced in the muscle as glucose 1-phosphate can only give the energy to the muscle. On the other hand, we can see the effect of glucagon and AMP on this muscle and liver glycogenolysis. Glucagon have no effect on muscle glycogenolysis, but they stimulate liver glycogenolysis. On the other hand, AMP stimulates muscle glycogenolysis, but it has no effect on liver glycogenolysis. Well, this was all about carbohydrate metabolism. I think this video will help you in your study. So please like, share and subscribe our channel. Thank you.